Hello and welcome back to the Biochemistry for Health Sciences channel. In the last several videos, starting from 5.0 to 5.29, we discussed various aspects of human metabolism. In today's video, let us summarize the most important points on human metabolism. One of the most important challenges the human body faces is to make sure that all the 30 trillion cells that are found in a human adult are able to generate energy at all times. Doesn't matter whether you are sleeping, you are awake, you are exercising, you are starving, you just had a meal, it doesn't matter. At all times, the cells, these 30 trillion cells, should be able to generate energy. For the 24 trillion cells in the body, these are red blood cells, to generate energy, they simply require fuel. Remember, red blood cells carry oxygen. So to generate energy, these cells simply require fuel. However, to most of the other types of cells, the remaining 199 types, most of these cells require not only fuel, but they also require oxygen. If we assume that there is adequate oxygen supply to these cells, so if we assume this is not a problem, then the problem becomes, how does the body provide fuel to all the 30 trillion cells at all times? Here is one of the most important limitations in human metabolism. This is a very critical limitation, and that is the brain relies very heavily on glucose as a fuel to generate energy. And since this glucose comes from the bloodstream, that means we must be able to maintain our blood glucose concentration greater than a critical concentration. And this critical concentration for humans is about 40 to 50 milligrams per deciliter. As long as we have more than 40, 50 milligrams per deciliter of glucose in our blood, the brain can extract this glucose and we should be fine. However, once our concentration of blood glucose goes lower than this critical limit, 40 to 50 milligrams per deciliter, what we call hypoglycemia, now the brain is not able to generate enough energy because it doesn't have enough glucose. Now we will suffer symptoms such as trembling, weakness, dizziness, seizures, loss of consciousness, maybe even coma and death. So this is a very important thing the body must do. It must always maintain blood glucose concentration above 40 milligrams per deciliter in order to feed the 0.1 trillion brain cells with fuel so that they can generate sufficient energy. So this is a very important question that we have to ask ourselves, whether the human has eaten a meal or is in starvation or exercising, doesn't matter what state the human is. How does the human being maintain his or her blood glucose concentration greater than 40 milligrams per deciliter at all times? There are three major metabolic states that humans go through. One is after a meal, postprandial. And then we have early starvation, that is between meals, for example, when you are sleeping in the middle of the night. And then we have late starvation, that is if we don't eat for more than about one day, about 24 hours. So let's summarize what we talked about in the last few videos. How does your body maintain blood glucose during these different metabolic states? Now, after a meal in the postprandial, assuming that we have a meal that has carbohydrates such as glucose, then it is your glucose from your diet It's a glucose from your diet that's going to maintain your blood glucose concentration. So since the blood glucose is coming from the diet, there is enough glucose, then all the organs, your liver, most important, your brain, the adipocytes, the muscle cells, kidney cells, and so on, all the cells in your body can use glucose. And if we have enough glucose in the blood, this is the only time when your blood glucose concentration goes much, much higher than normal. And therefore, that stimulates the beta cells in the pancreas to secrete insulin. And insulin basically stimulates synthesis. Okay, so in the liver, a lot of glycogen is synthesized. The non-essential amino acids are synthesized, leading to a lot of protein synthesis. There's a lot of lipids synthesized in the liver. In the adipocytes, again, a lot of lipids are made besides other biomolecules. And in the muscles, again, there's a lot of glycogen synthesized, glycogenesis, as well as there's a lot of proteins in muscles, so a lot of amino acid and protein synthesis happens, all due to the stimulation by insulin. Now, in early starvation, we are not eating anymore, so there is no more glucose coming from the diet. So now, where does the glucose come from that maintains blood glucose? And the answer is glycogen breakdown that happens in the liver, glycogenolysis, as well as synthesis of new glucose, gluconeogenesis. Under this situation, the insulin levels go down, and glucagon levels increase. So in starvation conditions, the glucose is reserved for the red blood cells and brain and a few other cell types, and the rest of the cells use fatty acids for their energy, for ATP. So the muscles use fatty acids, the adipocytes will use fatty acids, as well as the liver starts using more and more fatty acids, thereby reserving the glucose for the brain and the red blood cells. Now, if we do not eat for more than one day, then from early starvation, we will get into late starvation because most of your glycogen that is found in the liver that is maintaining this blood glucose, most of this glycogen would be gone in about 24 hours. Now, without the liver glycogen, now the question is, what maintains our blood glucose concentration? And the answer is gluconeogenesis, liver gluconeogenesis. Okay, this is the most important process that is going to maintain our blood glucose concentration so that our brain has enough glucose to generate energy. Besides liver gluconeogenesis, the small intestine also contributes to some glucose during this time. There's gluconeogenesis that happens also in the small intestine, and there's also a little bit of gluconeogenesis during this time that happens in the kidneys. So they also help contribute to the glucose concentration, maintaining the glucose concentration greater than 40 milligrams per deciliter during the early part of late starvation. So remember, gluconeogenesis requires energy. That's not a problem because the energy is coming from the fatty acids, from beta oxidation. But gluconeogenesis also requires building blocks. Okay, building blocks, carbon skeletons. And that comes from lactate and glycerol, as well as alanine. During this time, alanine that is produced in muscles. In this alanine, the nitrogen comes from the proteins, but the carbon skeletons, as we mentioned in the previous videos, actually comes from glycogen. This alanine, as well as other amino acids that are produced by protein breakdown, these all lactate, glycerol, alanine, and other amino acids can provide the carbon skeletons to the liver to make new glucose by the process of gluconeogenesis. So while the liver uses lactate, glycerol, and alanine, The small intestine uses mostly glutamine and the other amino acids to make glucose, and the same with the kidneys. It also uses, besides lactate and glycerol, it also uses glutamine as well as the other amino acids that are coming from the skeletal muscles. So this glucose that is being made now mostly from the liver and a little bit from the small intestine as well as the kidneys, this is mostly reserved for the brain as well as the red blood cells, while the rest of the cells once again use fatty acids for their ATP needs, for their energy needs. Okay, so the adipocytes will use fatty acids, beta oxidation. The muscles will use fatty acid, beta oxidation. The liver will use fatty acid, beta oxidation. The GI system, same thing, and so on. And so will be the kidneys. So that this glucose is reserved mostly for the brain as well as the red blood cells. So as the late starvation progresses, what we see is a slight change. So instead of the liver being the major source for gluconeogenesis, now what we see is a change. The kidneys become a major source of gluconeogenesis. Okay, so by this time the intestine is not contributing anything, while the glucose is still contributing to maintaining this glucose concentration in the bloodstream. Now, as you mentioned earlier, the kidneys use mostly glutamine as well as the other amino acids that are coming from the breakdown of proteins in the skeletal muscles. So mostly in the form of glutamine or other amino acids, as well as lactate and glycerol. These are all important building blocks for glucose synthesis in the kidneys. Now, another problem in starvation that the body should deal with is nitrogen, because breakdown of proteins means we are releasing a lot of nitrogen. And if the nitrogen is excess, then for the liver, when the liver uses proteins for gluconeogenesis, then the nitrogen is converted into urea, and then the urea is exported to the kidneys and comes out in the urine. However, in the later part, when the kidneys become the major source of glucose, or an equally important source of gluconeogenesis, then the kidneys will excrete the nitrogen, the excess nitrogen that's coming from protein breakdown, in the form of ammonia. And that ammonia combines with acid to give you ammonium. Okay, so the kidneys don't have to make urea, they can excrete the nitrogen in the form of ammonia. In the long term, breaking down proteins into amino acids and then using these amino acids as carbon building blocks to make glucose may not be a good idea, because muscle proteins, breakdown of muscle proteins is going to lead to muscle weakness. The person in starvation won't be able to move around too much. And then there could also be a possibility of breaking down some very important proteins in the body that carry out important functions. As you know, proteins do almost most things for cells. So this may not be a good idea, this continuous protein breakdown. So what we see in starvation, the later part of starvation, is we see the production of ketone bodies. Okay, ketone bodies are made in the liver. Basically, these ketone bodies come from fatty acids, and the liver now starts secreting ketone bodies into the bloodstream. And the most important thing here to understand is that the brain not only can use glucose, but it can use ketone bodies. So now the brain can use ketone bodies and glucose to generate energy. The red blood cells cannot use ketone bodies, so the red blood cells will keep on using glucose for its energy needs. The red blood cells and the liver do not use ketone bodies, but the other cells in your body, such as your GI tract, the brain, adipocytes, the skeletal muscles, the kidneys, all of them can generate their energy from ketone bodies. Okay, so this answer to this question, what maintains your blood glucose concentration? It depends on the state. Postprandial, mostly your dietary glucose. Early starvation, mostly liver glycogenolysis, a little bit by liver gluconeogenesis. And then in late starvation, no more glycogen left in the liver, so now we have to depend on liver gluconeogenesis, gluconeogenesis in the small intestine, and gluconeogenesis in the kidneys. So for the healthcare professional, it's very important to not just memorize metabolism, but to actually understand and apply metabolism. So please keep this big picture in mind, this very, very important point of how we maintain our blood glucose concentration during different times in the human body. So thank you for watching once again. Until the next video, goodbye and have a wonderful, wonderful day.